Hello everyone, this is Dr. Mahbubur Rahman. I am currently working as a research professor in Diosgenin University College of Medicine, Ligelia Cancer and Diabetes Research Institute. Today I am going to present my topic is that study for the mechanism of pain suppression by diosgenin by regulating the TRPV1 dependent signaling pathway. So if you see this title, so three main things. Number one is pain, and number two is diosgenin, and number three is TRPV1 dependent pathway. So this slide is this project is working on how diosgenin can regulate the pain through the TRPV1 dependent pathway. So first I want to introduce what is the diosgenin. Diosgenin is a phytosteroid. Saponin is the product of hydrolysis by acid strong bases or enzyme or saponin. This molar weight, this is the molar weight, and this is the chemical formula. So the main source of this diosgenin is it's originated from the plant. So Dioscorea species is like tuber like yam, and then Trigonella species, and also Smilex China. These three plants is the rich source of the diosgenin. So here actually this is, as I told before, this is mainly originated from the tuber like plant. So there are different types of tuber here. This also originated from the plant, and also from root and the seeds also. Trigonella from Trigonella seeds, and this is the root, Smilex China. The diosgenin is usually collected from this plant, but mainly is from the tuber collected from the tuber. So I previously published article. This is the Journal of Pharmacology and this work published in Journal of Pharmacology and Experimental Therapeutics Journal. So this in this work, they said the diosgenin modulate the vascular smooth muscle cell function by regulating the cell viability, migration, and calcium homeostasis. So this is the main, in this work, the main things is the diosgenin can regulate the calcium homeostasis. So from these interesting findings, I made the hypothesis how diosgenin can work on the TRPV1. So if you see this one, this work, in this published article, they found that diosgenin caused a marked decrease in the uridine triphosphate induced calcium transient in culture smooth muscle cells. So diosgenin can reduce if you see calcium current when use the diosgenin, diosgenin can reduce the calcium current flow. That means calcium influx. Diosgenin can reduce calcium influx. Here also they used another chemical, and they used it's like name is phenylephrine. They use this chemical in the smooth muscle cell, and then they used the diosgenin, and they also found that diosgenin can reduce the calcium current flow and calcium influx. You can see, however, in the presence of 25 microliter diosgenin, here also they use 25 micromole diosgenin. So another interesting article published diosgenin. Also in this article they said that diosgenin pre-treatment decreased the level of sodium ion and also calcium in the tissue. So these two works, I hear that diosgenin can regulate the homeostasis of calcium. So that is very important for the TRPV1. As you know, the TRPV1 is the one of the ion channel which regulate the calcium ion influx in the cell and thereby control the TRPV pain sensation. So now I am presenting that the evidence of Diosgenin can reduce the pain sensation in different model. So in this work, they found that Diosgenin can reduce the diabetic induced pain by diabetic neuropathic pain by the regulating the oxidative stress and the inflammation. So here also they found the another mechanism of Diosgenin, the NRF2 and HO dependent pathway. Diosgenin can modulate the diabetic induced neuropathy. So if you see this one, this also diabetic induced neuropathic pain, that Diosgenin can reduce the dose dependent manner the pain sensation in diabetes model. So here they use the conic constriction model, and also they found that Diosgenin can reduce the pain sensation and the heat hyperalgesia and the mechanical pressure by regulating the inflammatory and MAP kinase signaling pathway. So here in this article, they showed that Diosgenin can protect the neuron, and actually have neuroprotective effect through the regulation of autophagy pathway. You can see this one is BBB scoring is related to the locomotor and pain related parameter, this one, and they found that using the Diosgenin dose dependent manner can improve the locomotor activity in the spinal cord injury model through regulating the inflammation and apoptosis and autophagy related pathway. This one is also interesting. Is published on the Journal of Pharmacology journal. So this they found that as I told before, that Diosgenin is mainly collected from the Dioscorea species. There are different types of Dioscorea species, and Balbiferis L. barsativa is one of them. So Diosgenin can reduce the calcium channel. In potassium, they said that Diosgenin anti-nociceptive activity of methanol ester of this plant extract. So through the regulating of potassium sensitive potassium channel activation. So potassium channel is one of the cation channel which is also responsible for the pain regulation. So they found that Dioscorea plant extract, not the Diosgenin. I'm meaning that the Diosgenin is present in this plant. Maybe one of the influencing factor here is Diosgenin. So you can see that they use this plant extract, and then it's reduced the leaking time. So I will more Diosgenin also showed the analgesic effect in different. This is also plant extract. So with this plant extract, they found that this plant extract have the anti-analgesic effect. So my hypothesis is that the underlying factor is maybe Diosgenin. They didn't explain. So here this slide actually I I arranged because I just want to say that how the TRPV1 act in the pain sensation. So mainly the TRPV1 regulate the cation ion channel. This is divalent cation ion channel, monovalent and divalent. It regulate. So calcium is one of the main cation channel. It regulates. So through this one, TRPV1 ion channel, calcium enter to the cell and then increase the concentration of the calcium, and then this trigger the different mechanism like mitochondrial mitochondrial dysregulation, and also autophagy dysregulation, and also endoplasmic reticular stress, induce the endoplasmic reticular stress, and also apoptosis related signaling pathway. So the main thing is that the DRPV1 ion channel regulate the calcium ion channel, calcium ion influx in the tissue, the cell. So my research is that my experimental plan, so to show this one, actually this is my ongoing project, and this is I published on part of the project, and this my research plan, how I did. And this is the two part of this my project. One is in vivo and another one is in vitro. So in vivo model, in vitro model, we use the intracellular calcium measurement in HTRPV CHO cells, and DRG in the DRG cells. We use two types of cell, is CHO cells and also DRG cells. And another one is that in vivo model. So in vivo model, we use two types of pain model. One is acute pain model, and another one is chronic pain model. For acute pain model, we use the capsaicin induced pain model. We inject the capsaicin in the interplantar place, and we evaluate the behavioral test like pain like behavioral test. And also we use the CCI model, and then we inject, we administer the Diosgenin, and we evaluate how the change, and then molecular analysis is ongoing. We will evaluate this type of process. So this is our grouping, how we did this for capsaicin induced acute pain model. We use vehicle and Diosgenin and capsaicin, this one. And also for standard TRPV1, as you know, the TRPV1 antagonist is the BCTC. It's one of the sensitive and very good effective is antagonist is the BCTC. So we use this one for standard, and we evaluate how the Diosgenin can compare comparison with this one, with this drug. And also then how we inject, actually we used Diosgenin 50 milligram oral dose, 50 milligram per kg, and also 100 milligram per kg, and also BCTC we use 30 milligram per kg. And then we inject the capsaicin 1.6 microgram per pore, and then we evaluate how injection. Also we use the 400 micro this one 400 microliter vehicle. We use oral dose. First we used oral dose Diosgenin, and then after 60 minutes we use capsaicin in the interplantar flow, and we evaluate the pain related mechanism, and we make the score. And here also, if you see this one vehicle and capsaicin, so Diosgenin 25 microgram we use, and then capsaicin this one is oral dose, and this one is direct interplantar dose. So in interplantar dose we use vehicle 10 microliter, and after 30 minutes 30 minute capsaicin 1.6 microgram per 10 microliter. And also similarly Diosgenin, so we divide the Diosgenin dose. First half dose we inject, and then rest of the half dose we inject with the capsaicin, and then we evaluate how interplantar biting time or leaking time change. So if you see this one, this is the model, this is the leaking time behavior when we inject the capsaicin. So mouse feel pain, so it bite or leak to reduce the pain. It's like pain like behavior So first we inject, and then after 60 minutes, then we evaluate this one. And single, if you've seen, this is for oral dose. After 1 hour we inject, and then for single dose for capsaicin, 30 minutes we inject. So if you see this one, this one is for leaking time for oral dose. We evaluate just to make the video scoring, and then we also make the count the how many times it leaked the paw, how many seconds. And then if you see this one, when we used only capsaicin, the second is like 80 seconds. And then gradually when we use this low dose 50, and then when we use Diosgenin, and this one is BCTC. If you see this one, the similar like action in the BCTC. And also we used another experiment, we also used capsaicin, and similarly we used, and then we evaluate the Hargreaves test. It's like a heat sensation, and this one is for mechanical pressure. So time dependent manner, before injection of Diosgenin and capsaicin, we evaluate the how much pain it can tolerate for pain related behavior, pain sensation. We just evaluate, we hit sensation and how much it can tolerate. When it feel pain, then it just move, and then we record when it move the leg paw. And then if you see this one is like a 10 second, and then gradually it's decreased. After injection, only the capsaicin group reduce greatly, but Hargreaves can reduce. It's also feel little bit pain, but it's greatly reduced. At time dependent manner, it's improved in 2 hours, but only capsaicin group still feel pain in the the similar results also reflect in the bone frame mechanical stress. We again use, and we use bone frame, we use the fray, then we give the pressure, and then we found that we need to up feel pain, and then it moved the paw, and then we regulate this one. So after when we found that, if you see, when we found that the Diosgenin act very nicely, so we move for the next experiment. And you can ask why do we use capsaicin? Capsaicin is the agonist of TRPV1 ion channel. So, and BCTC is the antagonist of TRPV1 ion channel. So we used BCTC for standard, and then we use capsaicin for the to sensitize the TRP1 ion channel, and we use the BCTC to block the TRPV1 ion channel. And then we move for the experiment for you can see the capsaicin. We collect the calcium ion by TRPV1 in mouse dorsal root ganglion sensory neuron. We collect the dorsal root sensory neuron, and then in the cell culture we challenge that capsaicin. If you see this one, when we use the capsaicin, the current flow increased. That means action potential is increased. And then when we use the Diosgenin 10 microliter, is greatly reduced. If we compare these two peak, you can see this Diosgenin also reduced. So we hypothesize that from this result is reflect that Diosgenin can directly reduce the TRPV1 ion channel, act on TRPV1 ion channel. And after that, we also evaluate the CHO cell, CHO K1 cell. This one is we used, and then we used the 1 microliter of Diosgenin, and then also we found the capsaicin. You can see this if we compare these two peak, and here how it's significantly increased, and this one after Diosgenin is reduced. So it's also showed that it proved that Diosgenin can reduce the TRPV1 ion channel, block the TRPV1 ion channel. As you know, the TRPV1 ion channel have the big problem is that after it's not clinically used because of it has the temperature. It make problem in the temperature of the body. Some TRPV1 ion channel greatly increase the TRPV1 ion channel. Most of the TRP1 antagonist increase the body temperature, hyperthermia, induce the hyperthermia, and some part reduce the hypothermia. So in BCTC, in rat is in mouse, rat and mouse, mostly it increase the temperature, hyperthermia, induce the hyperthermia. So we compare that how the Diosgenin can reduce the temperature or not, or Diosgenin is now it's proved that Diosgenin worked on TRPV1 ion channel, but we want to see that is it work like in body temperature like the BCTC or not. Then we evaluate this one. We injected them BCTC and also similar dose of Diosgenin, and then we evaluate the rectal temperature with this thermometer, digital thermometer, time dependent manner. And then we found that after injection, BCTC temperature is increased significantly, but the Diosgenin have no significant effect. So it's very nice. So then we move for the next experiment, next chronic experiment. So we induce the CCI induced chronic constriction injury and sciatic nerve surgery. We make surgery, and then vehicle we use 10 mice, and then if you see this one CCI, and after 3 days Diosgenin 50 milligram per kg orally once daily, and then this one 100 milligram per kg once daily, and CCI, and after 3 days 30 BCTC. We also inject BCTC for BCTC, and after 3 days 30 milligram per kg orally. First we induce the CCI, and after we wait until 3 days, and then we start the treatment. If you see this diagram, how we use the we measure the BCTC, this the green color, and then we make surgery. After evaluation, the pain tolerance level and by the heat sensation and the mechanical pressure in the zero, and after the measurement, the level, normal level, then we make the surgery, and then we measure after 3 days. We measure this one, but we give the treatment continuously, and after 3 days we found that you can see 1 to 3 days. Until 3 days we didn't treat anything. So until 3 days we again evaluate the pain level, and then when we confirm that the CCI model induced the pain sensation and chronic pain induced, and then we move for the treatment, how Diosgenin can improve the pain condition, or how it can reduce or elevate or attenuate the pain sensation. And then from 3 days we started the treatment, and after until the 9 days, and 10 days we sacrifice the animal. And then if you see, this one is how we make the CCI model, chronic pain sensation by surgery. And we also measured the body weight, and we found that the pain sensation have lower body weight, but there is no significant change throughout the experiment. But at the end, the body weight, the CCI model body weight was lowest, and the treated group body weight also is like almost like normal. And the poor withdrawal latency, if you see this one by Hargis test to evaluate in the by time dependent manner, and the treatment of BCTC and also Diosgenin gradually improve the heat sensation like normal. And also if you see this one for the mechanical pressure, it also increase. So then we collect the sample, we measure. This project is ongoing, but we just published this one part of the work. For mechanical pressure, if you see this, we use the for the molecular experiment, molecular mechanism. We measured the anti-inflammatory pro-inflammatory cytokines, and TRPV1 ion channel. So we found that in CCI model, if you see this one, TRPV1, if you see this one, TRPV1 sensation increased and the treated group decreased significantly. If you hear this one, and then also in pro-inflammatory cytokine, it also reduced. So at the end, so we will strive for more molecular mechanism in the to identify how it's correlate with the digestion, how TRPV1 and other molecular mechanism can regulate. So we will move. We are ongoing this ongoing project we are doing. So until this we already we've already published. So we already published. You can find this article by name Mahbubur Rahman and Diosgenin. You can find the published article. So in conclusion, we want to say that our results suggest that Diosgenin exert the analgesic effect and reduce the chronic inflammation by antagonism of TRPV1 in a mouse model of pain without altering body temperature. So no body temperature change. So this is the main thing, that TRPV1, the proposed TRPV1 antagonist make the temperature problem, induce hyperthermia, some induced hypothermia. So but this Diosgenin didn't change significantly in the body temperature. And also this current study provides the new insight of mechanism underlying the pharmacology effect of Diosgenin, which may in turn provide insight into the therapeutic effect. So Diosgenin may have also value to reduce the risk of long-term inflammatory complication in patients with chronic neuropathic pain by a TRPV1 ion modulation. So therefore, Diosgenin represents effective therapeutic agent and in acute and chronic neuropathic pain disorder in mice. So this is my project. This is today I want to present until this one. And if you have any question, you are welcome. You can ask. I provide my personal email. I will try to give the answer. Thank you.